The next, before translation, we have to understand the basic structure of transfer RNA. So in the transfer RNA having 70 to 90 nucleotides, and it's a secondary protein structure, and it's a clover leaf pattern type of RNA clover form. So how a clover leaf looks like: we have a long acceptor arm and a short arm. Then we have a D arm and a T arm, and then we have an O type structure or anticodon type structure. So D, T and O. So in the T section we have cytidine, pseudouridine and thymidine, ribothymidine. In the D side we have two structures, dihydrouridine structure. So D site is this. The next is the anticodon site. Anticodon site is complementary to the messenger RNA, and it has complementary to the it has the base sequences which are complementary to the messenger RNA. This is the messenger RNA came out of the nucleus into the cytoplasm, and now transfer RNA is now going to bound it in order to produce protein synthesis. So this is a relatively clover leaf like structure. Over here we have a variable arm, non-specific. So this is actually the main structure of a clover leaf pattern of transfer RNA. This is the 5' end and this is the 3' end. The 3' end, we have the three aces, and these aces are the acceptor site for the amino acids. So amino acids come and covalently bound to this site. So this site has the highest percentage of modified. The whole sequence has the highest percentage of chemically modified bases in it. The next we have the cytidine. We have uridine, and this uridine is pseudouridine, and we have the thymidine, and this is attached to RNA, so it's a ribothymidine. And this T for thymidine, T for T arm, and T for tether. Tethering what? Tethering mean binding. It tethers to T arm, tethers to the ribosome. Then we have opposite to this acceptor site is the anticodon site. This is a anticodon arm or anticodon site, and it is complementary to the messenger RNA. The next we have the D arm, and this D arm has enzyme. Which enzyme? Synthetase enzyme, transfer RNA synthetase enzyme. So one synthetase enzyme is specific for one pair of amino acid and transfer RNA. And this synthetase enzyme actually it recognize the transfer RNA in its amino acid. It matches the amino acid to the transfer RNA. It scrutinize or proofread whether or not a correct amino acid is attached or not. And this synthetase enzyme need ATP. So whenever there is amino acylation or protein charging, the transfer RNA is charged. So this enzyme is very much important. If this enzyme is if we have the problem with this enzyme, like in the polymyositis or dermatomyositis, which are rheumatological condition, connective tissue disorder, dermatomyositis, we have antibodies against the antihistidyl, which are modified histone trans RNA synthetase, which leads to this autoimmune condition. So this is the general structure of transfer RNA. We have a clover leaf pattern, we have 3 arms, how they interact, and how if another thing, if there is a mismatch amino acid is mismatch transfer RNA. If a mismatch amino acid, it get charge on it, so mismatch transfer RNA it read correctly, so reading is not impaired, but it inserts a wrong amino acid into the protein growing chain. So this is a brief introduction of transfer RNA and how it has different components and it's different. So this transfer RNA, this synthetase get ATP and charged, and this ATP and this amino acid which is covalently bond with this site, it's now it has a shape like amino acid having this, it bound to, and it now goes into the messenger RNA over there. It give the amino acid and then release off. So for start codon we have the AUG or as a mnemonic inaugurates amino acid synthesis or protein synthesis, and for termination we have the release factor. We have initiation factor or we have the release factor, and release factor recognizes the stop codon, which are the you are away, you go away, you are away, now you are gone, you go away, UGA, UAA and UAG. So these are the stop codon. So AUG is the start codon. So this is the brief introduction of transfer RNA.